Hi there, ladies and gentlemen, Bio 400 students. This is Mr. Workman.
And Mr. Gales.
Today we're going to bring to you your first screencast session. We're going to be going over the scientific method and the process of designing experiments.
Well, this is going to be broken down into two separate screencast sessions, and in the first session we're going to focus on the following topics. We're going to be looking at the goal of science, which is really why we do science. We're going to learn about the steps of the scientific method that would be commonly followed. We'll describe what a hypothesis is and how to write one. And then we're going to look at the independent and dependent variable and how to identify those. That'll be screencast session 1.
And in session 2, when we look at the scientific method and experimental design here, we're going to go more into experimental design and how sample size and validity of your results are connected. We'll talk about how data collection and organization of that data can be accomplished. And then following that, we'll work through what conclusions and theories are. And then we're going to summarize, or end up, I should say, with what the characteristics of science are, what science is and what it is not, so to speak.
All right, so let's get right into this by looking at science. And before we move into our first topic, which is the goal of science, we're going to have a fairly well-known scientist describe for you why he thinks science is so important. This is Neil deGrasse Tyson talking about why we need to learn about science. And so as you watch him describe this, the importance of science, just kind of keep that in the back of your mind. And as we go into the goal of science, we'll reiterate some of his points.
Listen. Science is all around us. It's in us. Knowledge of science is power. It gives you an understanding of the forces of nature, which some people will exploit your ignorance of it if you don't know it. Hey, I got some crystals here. If you rub them together, they'll heal you. Ah, you might say, well, what are the crystals made of? What diseases do they cure? Where did you get them? How much do they cost? Why do they work? You start bringing forth that series of questions, the person runs away in tears because you know how to ask questions about the natural world. It's not even about how much science you know. It's about how science literate you are. Science literacy is brain wiring. It's what's your first thought when someone tells you something? Are you in a position to know whether they're just full of it? Or are you positioned to say, hey, that'll work and I know why? The laws of physics, chemistry, biology, they allow it. They thrive on it. So, I say to you, if you're one of those who says, well, that's science, that's not me. Science is you, and the sooner you know it, the more empowered you become. Science literacy is a vaccine against all the fuzzy thinking that goes on in the world. Why not get vaccinated? Therein is power. Signing off from Kennedy Space Center, Florida. Behind me, the International Space Station, the member countries. This is Space Coast. Neil deGrasse Tyson, signing off.
All right, so as we look at science here, and as you noted, I'm sure Neil deGrasse Tyson described how important science is and why it's important. You need to really understand that science is really a process. Most of us, or most people, think of science as a noun, a thing, but it's really a verb. And that process is based on observations or experimentation, and of course, through a result of experiencing your observations or going through your experiments, those provide explanations of natural events or particular problems that you may encounter as a scientist.
And as you saw in the video, he talks about being vaccinated against all the misconceptions that go around in the world around us all the time. Science is really a way that helps us to understand the natural world in what's called an empirical way. What that means is that it relies on the evidence that we can gather by using the tools of science and the scientific method. So it really is focused on gathering evidence about the way nature works.
And because of that, our conclusions, or what we think we need, what we think we learn or come to new understandings about as a result of doing science, so to speak, it relies on what we call facts or data. This could be numerical, or it could be informational or descriptive data, but it relies on verifiable data.
All right. Now, our next topic that we're going to be looking at here, and by the way, I should mention this as we're learning how to do screencast notes. Anytime you're seeing a bolded term, and we're going to go through this together in class as well, anytime you see a bolded term that's underlined, that'll indicate to you that that's a main idea and should go on the left. So science would be your main idea on this slide here. I'm going to go to our next slide. The main idea on this slide is the steps of the scientific method. So you would write steps of scientific method on the left-hand side of your paper. And then on the right-hand side, you're going to take your notes, which in this case is a numbered list. Notes that you would do on the right-hand side would include definitions. They could include, listed information like you see here. They could include examples. If there are pictures involved that you think might be important, you would want to include those in your notes. And also any questions that come up is an important thing for you to have in your notes as well. So with that information having been said, let's look at the steps of the scientific method. I want to note that not every scientific experiment will follow every one of these steps, but this is a general outline for how we would go about doing science. And it begins with making observations, using your senses to gather information about the world around you. And when you've done that, when you've been observant, a lot of times that will bring up questions or problems that you may want to have some solution to. So the next thing that you would do, once you have your problem stated or your question, you need to do a little bit of background research so that you can get educated on your particular topic. And after doing that background research, you're able to move forward with the next step of the scientific method.
So once you've got your research and a question asked, what you're going to try to do is make some sort of prediction or link why certain things happen, what causes what effect. And that is essentially what a hypothesis is. It's an initial explanation of two linking variables. And following that, you're going to experiment or continue observations to verify whether or not your hypothesis is valid, or if potentially your hypothesis could be refuted based on your experimental data or subsequent observations. Once you do your experiment, of course, if you do have data, you need to collect it and organize it in some sort of way so that you can then analyze it and make some sense out of it. Following that analysis, you're going to interpret your data and hopefully be able to draw some conclusions from those data, or that data set, I would say. Now, one of the great things about science is that it's refutable. So if I do these 7 steps and somebody else wants to try to verify or maybe invalidate my claim, we could go through these series of steps again and retest it. And if it happens to be good and valid research, and another set of scientists could critically review my work, if it seems significant and valid, then we might go and publish what I've learned as a result of this process.
I think it's important to note, people oftentimes ask why would scientists want to publish their work, and the easy answer to that is it is the source of all we know about the natural world. There are really 2 major reasons why we do science. One major reason is what's called pure knowledge. It's just understanding the world around us. And the other reason is to develop technology. Technology, in essence, is applied science. So that publishing step is very important. Now, before we move on to the next slide, this silly little cartoon picture here actually is a pretty good flow chart for how the scientific method works. And notice, once we've gotten to the point where we're ready to do our experiment, we're actually doing hands-on, we're going to look at the data. If our observations and our experimental data does not support our hypothesis, you need to consider, is there something that you did incorrectly, or you might want to design some different experiment to go about testing that hypothesis. Obviously, if your data supports it, then your conclusion is going to be validated, or your hypothesis would be validated, and then you go on to communicating your results. All right, our next main idea is the hypothesis.
So again, here's your main idea, hypothesis. Put that on the left-hand side if you're doing your 2-column notes here, which you should be doing. And on the right-hand side, there's the information pertaining to that main idea. And here's your definition, of course. A hypothesis is a possible explanation of a natural event or an observation that you've made that could be tested by either subsequent or additional observations or experiments that you're going to design and then run. The key thing about a hypothesis is that it must be testable.
Yeah, if you look at this picture here, you've probably experienced where you have a house plant that gets wilted. It's not standing upright. It doesn't look very healthy. Let's say you come up with some completely off-the-wall explanation for why this house plant is like this. Like, for instance, space aliens came down and did something to it and made it wilt. That's not a good scientific hypothesis because it's simply not testable. It would be a fun explanation, but it's not a good hypothesis. It has to be something that you can practically manage given the resources and the space that you have, the time constraints, the money restraints, and so on.
So if I said something like, I think the flying spaghetti monster came down from the heavens and sucked all the plant juice out of this plant, would that be a testable thing, Mr. Gales?
No, that's not verifiable. It's not testable in the sense of true science, so no.
All right. Well, let's get into a valid scientific hypothesis then. Very frequently, what we're going to see in our hypotheses, which is the plural form of the word hypothesis, is that they are written in an if-then statement format. And the reason for that is we're going to try to link what we call an independent and dependent variable, which we're going to go into later. But we need to say that something we're seeing is the result of a particular cause. And we're going to either observe or test or experiment to see if we can get that prediction or that hypothesis statement to be verifiable.
Now, to help you understand how these important variables, the independent and dependent variable, are linked to the hypothesis, we're going to watch here just a quick short video about a science student who's helping his teacher out with an experiment.
Hey, good morning, Mr. Koenig. What are you doing?
Good morning, Jacob. I am doing an experiment.
Well, do you mind if I help?
Of course you can, Jacob. Come on over here. I'll show you what I'm doing. Look up here on the windowsill. I have two plants. Both the plants are in pretty much the same location and get the same amount of sunlight. This first plant, however, I water every single day. This plant, however, I only water every other day. So, Jacob, in this experiment, what do you think it is that I'm testing?
Well, since they're the same type of plant and in the same location, are you testing how well they grow depending on how much water they get?
That's absolutely correct, Jacob. I'm measuring how much the plants grow based on how much water they get.
So why did you place them both in the same place? Wouldn't it have been fun to put one somewhere and somewhere else, and maybe have different amounts of soil? Oh, we could also change what music we play for them.
Easy. Calm down, Jacob. That actually wouldn't work in this experiment. Let's talk about why. If we set up the experiment in one of the ways you suggested by putting the two plants in different locations, this first plant would receive more water but less sunlight. The second plant would get less water but more sun. So do you see the problem yet, Jacob?
Oh, what, Mr. Koenig? I think I do. We're changing too many things. At the end of my experiment, I couldn't say that it was the water or the sunlight that made it grow more, because I changed too much.
You got it, buddy. In order for an experiment to be fair, you can only change one thing at a time. Everything else in the experiment should stay the same. In your experiment, the thing that you change is called the independent variable. In order for a test to be fair, you must always only have one independent variable. And remember, that's the thing that you're going to change. In our case, it was the amount of water that the plants received.
You know what? I think I get it. All of those things that I talked about, like changing where they were and the music that they were listening to, if I selected just one of those, they all could make good experiments. But I can only select one.
That's right, Jacob. And the one thing you select becomes your...
independent variable.
Right you are, Jacob.
So, Mr. Koenig, is there a name for everything else?
What do you mean, Jacob?
You know, like all of the stuff that we could have changed, but we didn't. Like keeping the plants in the same location, or letting them have the same amount of light, or using the same kind of plant.
That's a great question, Jacob. And yes, they do actually have a name. They're called the controlled variables, or sometimes just the control. The control, or the controlled variables, are the things that stay the same in the experiment. The point of a control is to keep an experiment fair. By keeping everything the same and changing just one thing, we can say that that one thing did or didn't have an effect on what we were testing.
Wow, very cool. So is that all there is to an experiment, Mr. Koning?
Nope. When it comes to scientific experiments and variables, there's just one more part. But let's look at that after spring break.
Sounds good to me, Mr. Koning.
All right, so we're still working here on hypothesis as a main idea. And as you saw from that short video, the student and the teacher were attempting to try to link two variables, what is causing what. And the first thing that I'm going to go through here with you is what we call the independent variable. And of course, what that is it's a factor that's deliberately manipulated in an experiment. And if you remember the video, what the science teacher manipulated was the amount of water that was being given to the two different plants. You can think of the independent variable as the presumed cause. The dependent variable depends on the independent variable. You can think of that as the response, or maybe the presumed effect or the result of the independent variable. So this graphic that's here on the right, cause, independent variable, effect or outcome, or sometimes thought of as the result, is your dependent variable. And your hypothesis is usually written as if independent variable, then dependent variable.
Couple things to think about here. One, the independent variable is oftentimes just simply referred to as the experimental variable. For our purposes, we really want you to work with independent variable. It's the factor that you manipulate. Think about it this way. It's what I change. If I'm the scientist, it's what I change. I, independent. All right. So let's take a look at an example of a hypothesis and how we use the if-then format to identify these variables. Here you can see that there's a hypothesis, and we've talked about the if independent variable, then dependent variable. And from the video, right, we would look at if a plant is watered every day, then it will grow more than a plant of the same species that is watered every other day. If we have a hypothesis that's written correctly, we should be able to look at it and identify that cause and effect. The amount of water in this case is the presumed cause. The amount of growth would be the presumed effect. So getting the hypothesis written correctly is a critical part of doing science properly.
So how about you try this really quickly? Let's set up this particular example. And this is an example hypothesis. What I want you to try to do is identify within that statement what the independent variable is, and then also what the dependent variable is. So here's your statement. If 100 grams of fertilizer is applied to a lawn, then the lawn will grow at a faster rate. Pause the video, take a moment, see if you can isolate which variable is which from that statement.
All right, let's try this. Remember that a hypothesis is written if independent variable, then dependent variable. So if 100 grams of fertilizer is applied to a lawn, if you guessed that the independent variable would be the amount of fertilizer, then you're correct. The amount of fertilizer is what you as the scientist would change or manipulate. The dependent variable would be how fast the lawn grows, the rate of lawn growth. And we'll talk about in the next screencast, we're going to talk about data and perhaps different ways that you might measure the growth of plants, for instance. But this would be a simple example for you to go through. Now, here's something important when you're watching screencasts. If there's something that you didn't understand, make sure you write that down in your notes, because we'll have an opportunity in class to talk about those questions. If you did not understand how to identify the independent variable and the dependent variable from the hypothesis, make sure that you address that in class with us. All right, this is the end of Screencast Session 1. You should have your notes completed on this, and I would encourage you to go back and maybe watch the video one more time without worrying about taking notes, just to make sure that you're processing it. Remember that if you have any questions, that when you come into class, we're going to be going over, spend a little bit of time going over that information, and then you'll be eventually tested over that as well.
Thanks, everybody. This is Mr. Workman.
And Mr. Gales.
See you next time.